Hello. Thank you so much, Melanie. Thanks for organizing, and thanks for everyone who helped organize the conference, and thanks for you guys for staying here till the last session. Um, okay, I guess I'll start. So my name is Abdullah Syed. I go by my last name, Syed. I'm a postdoc in Jennifer Doudna's lab at the Gladstone Institutes, and I've been working very closely with Melanie Ott's lab for this whole project. And basically, what I'll be talking about are virus-like particles, which you heard about earlier with Rebecca's talk, with a slightly different twist. So in particular, I'll be looking at the N protein phosphorylation on SARS-CoV-2 assembly and replication. And going to the next slide. So before I get into it, the goal of this project was I wanted to study the assembly process of SARS-CoV-2. So this is the full infectious cycle. And roughly, I'm considering all of this part after entry to be replication. And then after the proteins that are required for making the next generation of particles are produced, these proteins have to assemble, encapsulate the viral genome, and export it. And I'm considering this part to be the assembly process that I really wanted to understand. So the key proteins that make up the SARS-CoV-2 virion are the spike, E, M, and N proteins, shown schematically here. Everyone knows the spike, so I don't think I need to introduce it. But the E and M proteins are key for assembly, the M protein forming the envelope, and the E protein with a number of other somewhat poorly characterized functions. And the protein I'll be focusing on more is the nucleocapsid, or N protein, which forms a helical capsid around the genomic RNA. So shown here is the only protein that's not transmembrane, embedded inside the particle, and it packages and protects the RNA. And basically, what I ended up developing was a way of making virus-like particles that encapsulated an exogenous transcript. So the original SARS-CoV-2 has these 4 structural proteins and the RNA genome, and I wanted to substitute the RNA genome with a reporter transcript along with the packaging signal. It's a slightly different strategy than what Rebecca was talking about, with either encapsulating the protein or using a different packaging signal and a different capsid protein from a different virus. But I wanted to find the packaging signal of SARS-CoV-2 and use that to encapsulate exogenous transcripts. And for the sake of brevity, basically the sequence I use, I've turned to T20. It's found in the 3' end of the first open reading frame. So SARS-CoV-2 and other coronaviruses have one very large open reading frame, and near the end of it is a sequence that serves kind of as a packaging signal for SARS-CoV-2. The sequence had not previously been characterized. So what I helped develop was this method for basically generating these virus-like particles from transfecting the core components. So if I take human cells, 293Ts in this case, and co-transfect with the 4 structural proteins, along with a transcript that contains a packaging signal. So in this case, I'm using luciferase. When I co-transfect these proteins, the cells produce virus-like particles. And then these virus-like particles can be either purified or filtered and then added to cells that have the entry factors of SARS-CoV-2, and this generates luminescence. And so I'm very lucky that I chose firefly luciferase and not nanoluciferase, as you heard from earlier. So initially I had a lot of problem with getting any signal at all, because as you heard, firefly luciferase does not get non-specifically packaged. But eventually we figured out a system of making it work, and when we have all 5 components, we get luminescence. And if we take out any one component, or if we leave in the luciferase transcript but remove the packaging signal, we get much lower levels of luminescence. And we also see transient luminescence in the infected cells that fades over time, in comparison if you use a lentiviral vector, where you get expression. The expression increases over time, as you would expect. One moment, sorry. So there's a number of things you can do with virus-like particles. As you know from other work that has been presented today, they can be used for drug screening, for understanding assembly, for a number of other things. What I wanted to look at initially was just some of the mutations that were happening rapidly at that time. And I'll be talking a little bit about the nucleocapsid mutation especially, We looked at mutations in all the structural proteins, but the emerging mutations in nucleocapsid protein, and we could do that very easily by just making the mutations in the plasmids, co-transfecting them, generating VLPs, and seeing what impact they had on the phenotype. So I did this in close collaboration with Melanie Ott's lab, and especially Taha, who's sitting in the audience as well. And before I tell you the results, even before testing it, we found an interesting relationship where a large portion of the mutations in the nucleocapsid protein seem to occur in the 7 amino acid region of the nucleocapsid protein. So the nucleocapsid protein has 2 well-folded domains, often called NTD or CTD, NTD and CTD, or N-terminal domain, C-terminal domain. And then what I'm showing as thin lines are intrinsically disordered domains, and the one in the middle is called a linker. And in the middle of a very flexible serine-arginine-rich region, there was independent mutations in all the variants that have shown up since the beginning of 2021. And not only that, when we created those mutations in virus-like particles, we found that the virus-like particles were 10 times more efficient at packaging and delivering luciferase. So we characterized this thoroughly. What we found was basically that these mutations don't increase the amount of particles that are produced, but they increase the efficiency of RNA being packaged and delivered within these particles. So potentially, without these mutations, the same number of particles are produced, but they're lacking as much, or many of them are empty or hollow, and then with the mutations, they're better at capturing the specific RNA. Again, with help from Melanie Ott's lab and Taha and Takako especially, we found that these mutations in BSL3 in infectious clones enhance replication by up to 3 logs. So these are real beneficial mutations for the virus, but there was no other system at that time that could really characterize why these mutations were occurring and why they were helpful. And since then, I've been trying to understand what effect these mutations have. Sorry, I'll skip these slides for brevity, but looking closely at where these mutations occur. So this is actually the sequence where these mutations are occurring, and on the bottom I've labeled what these mutations actually are. They occur in a region that's heavily phosphorylated by an enzyme known as GSK3, and many of the mutations are either directly disrupting phosphorylation or are immediately adjacent to sites of phosphorylation. And previous work from other coronaviruses has shown that this phosphorylation is very important for nucleocapsid assembly into either tight particles or into phase separation. So we were wondering whether this could be the reason why the virus was enhancing its ability to package. And so I worked with David Morgan's lab at UCSF, and what he was able to show is that these mutations, in fact, reduce the amount of phosphorylation in this region. Compared to the wild type, nearly all the mutations that have shown up reduce the phosphorylation substantially, but not to 0. They don't completely eliminate the phosphorylation, but they reduce it substantially. And if we block GSK3 using an inhibitor, in this case CHIR98014, what we find is the mutants that were already very efficient, they don't seem to respond to this inhibitor, but the wild type and other mutations that don't respond to this inhibitor, or sorry, that don't affect the phosphorylation, get much more efficient at packaging with the added drug. So basically, it seems that the original wild type had too much phosphorylation for efficient assembly, and then reducing that phosphorylation led to more efficient assembly. So we're still working on exactly why that is the case. So here's further data. So if we generate specific mutations, so the way GSK3 phosphorylation works is that there's a priming kinase that phosphorylates at 206, and then GSK3 phosphorylates every 4 amino acids upstream of this site. So if you block the 206 phosphorylation, you'll block all 5 of these kinase events. And so substitutions at 206 generate 20 times more luminescence, and then at 202, about 10 times, and then at 198, about 3 times. So progressively, as you reduce phosphorylation, you get better assembly. So we're still exploring, why this phosphorylation is affecting the packaging ability. But so far we see the same effect also in live virus. Here, we're comparing the Washington isolate, which has no mutations, or the wild-type nucleocapsid sequence, versus the delta, which has one of the mutations that were reduced phosphorylation and enhances assembly. And what we see is the delta does not respond to the inhibitor, the GSK3 inhibitor, whereas if you add the inhibitor to the Washington isolate, you get much higher assembly. So what I'm continuing to work on is, why does N protein get phosphorylated at all? What is the normal physiological function of it? It has been shown previously that it controls phase separation and affects how the nucleocapsid assembles. But we've been able to show that even eliminating phosphorylation at these sites still allows assembly of these virus-like particles. And it's also not clear why phosphorylation blocks packaging. So why does it exist? Why does it block packaging? And one of the hypotheses that I think is happening is perhaps human cells have more GSK3 activity and higher levels of phosphorylation than whatever the original host was for SARS-CoV-2. And that's it. Thanks so much for listening, and thanks especially for Melanie Ott's lab. Several people, Taha, Takako, and Irene, and my research associate, Allison, who did a lot of these experiments, and Jennifer, who's my advisor. Thank you.
Just a real quick question. When you applied the CDK3 inhibitor at higher concentrations, just double checking, did you check for cytotoxicity to see if it was causing the cells to die and that's why you're losing your effect?
Yeah, so it is. So can we put the slides back on? Okay. But basically, at the very highest concentrations, there is cytotoxicity, and in that case, no matter the variant, you get reduced efficiency. But at lower concentrations, it's non-toxic. And actually, at the 5 micromolar concentration, this drug is actually commonly used for maintaining stem cells, for induced pluripotent stem cells. So it's a very commonly used drug at the lower concentration, where it has a very potent effect.
Carla?
Hi. So when I first saw your work, I thought that maybe the phosphorylation, the dephosphorylation was necessary for the switch from RNA replication to packaging.
Yes, I left out some of this data just because I wanted to make it shorter. But yeah, so I've been working on figuring out whether, so one of the earlier hypotheses was that the phosphorylated N protein would be better at replication, and the reduced phosphorylation would be better at packaging. And as I dig deeper into it, I'm not sure if that's still the case. So we've been testing that with a replicon system, and it's really hard to control, to separate the assembly and packaging steps, completely separate those two. But as I'm getting better at separating those, it seems that the phosphorylation is not really helping the replication step either. So I'm no longer convinced that it's working as a switch and may not be required at all.
Well, it's got to be there for a reason.
Sorry?
It's got to be there for a reason.
Yeah, it does. I'm still trying to find out what that reason is.
Very cool. Thank you.
Thanks.
Syed, I can't remember if I've asked you this before, but the packaging site that you've sequenced that you found, is that conserved across other coronaviruses? Is it that you're, and then the second question is I don't remember if you showed this in your talk, but do you see any changes to the level of phosphorylation of N in a kinetic sort of sense over the time course of infection? Is it sort of regulated in that it accumulates phosphorylation more at the beginning, one would assume, rather than at the end if it's inhibiting packaging?
Thank you for both those questions. Yeah, I'm working on the time-dependent phosphorylation especially, but I don't have the data yet. But in terms of whether it's conserved, so it's conserved in lineage B beta coronaviruses, but it's missing, or they're different. That site does change quite a bit as you go further out from that family. So I'm planning to test SARS-CoV-1 and compare those packaging sites soon. But it is not completely resistant to mutations. And actually, I've tried making mutations that don't really seem to have that much effect to it. So it seems to be like it's not that sensitive to the exact sequence. And one of the reasons is because it's still relatively large. I've narrowed it down to about 850 nucleotides, but that's still quite a bit of sequence that, you know, could have complicated structures and maybe, you know, somewhat tolerate some mutations.
Well, thank you very much.